Hello, good morning. Today we are going to see what exactly is protein binding of drugs, how protein binding occurs, reasons for protein binding, what exactly we will see in detail in this module about protein binding of drugs. So myself Prashant Nayak, Assistant Professor, Department of Pharmaceutics, NGSM Institute of Pharmaceutical Sciences, Mangalore. So our topic, the learning objectives of our topic. First, we will give a general introduction on what exactly is protein binding, how it occurs, what are the main elements which are present in it. Then we are going to learn about mechanism of protein binding, how the protein binding occurs in the body, what is the mechanism, what are the main role players, and then we are going to see how to determination or how to evaluate the protein binding of drugs. So these are all things we are going to learn: introduction, mechanism, and how to evaluate the protein binding of drug has taken place or not. So we'll see one by one. Introduction to protein binding. A drug in the body can interact with several tissue components, of which two major categories are: one is the blood and one is the extravascular tissues. So the interacting molecules are generally macromolecules such as proteins, DNA, adipose tissues. The proteins are particularly responsible for such an interaction. The phenomena of complex formation with protein is called as protein binding of drugs. Protein binding is divided on intracellular binding and extracellular binding. Extracellular is the one which is before the cell, and intracellular binding occurs when the drug enters inside the cells. So the protein molecule inside the cells, when it interacts and the binding takes place, we call it as intracellular binding. The binding in which it happens during the blood or into the intracellular fluid, we call it as intracellular binding of drugs. So protein binding divided by 2. Now, mechanism of protein binding. During the protein binding, what exactly happens? Binding of drug to protein is generally reversible, which suggests that it generally involves weak chemical bonds. They are always reversible. Protein binding, protein drug binding. There can be hydrogen bonds in which the protein and a drug is bound together with the hydrogen bonds. The bonds are generally weak and they can be reversible. Hydrophobic bonds which are present in the environment, they will have combination on protein which are the bonds hydrophobic, and the drug will attract towards it and form a hydrophobic bonds which are also generally a weak bonds. Ionic bond occurs. Ions which bond together from both the protein and drug, they combine together and bind, and van der Waals forces of attraction. So these are the various mechanism of protein drug binding. It occurs because of hydrogen bonding, because of hydrophobic bonding, ionic bonding, and van der Waals forces. So it generally involves a weak chemical bond, and the bond is always reversible. Irreversible binding of drugs, though rare, arise as a result of covalent bonding and is often a reason for carcinogenic or tissue toxicity of the drug. Covalent bonding of chloramphenicol paracetamol metabolites to liver results in hepatotoxicity. So, reversible as well as irreversible. Irreversibles are very rare, but it occurs a covalent bonding always. When this occurs, there is a severe toxicity of the drug or carcinogenic effect seen. Example of paracetamol, which causes hepatotoxicity. Binding of drug falls in two classes: binding of drug to blood components like plasma protein and blood cells, binding of drug to extravascular tissue, proteins, fats, and bones. These are the second one class. Now we will see in influence of binding of drugs and its clinical response. What exactly happens in figure 1? which is taken from the reference number 1 and 2. Now coming to the first picture over here, liver, drug and metabolite binding to liver tissues. The protein drug, a drug metabolite forms binding of liver tissues in the liver. Then the drug is along with the enzyme forms a metabolite. The metabolite in turns forms protein drug complex. This goes, metabolites always go to biliary excretions and get eliminated. Now starting with the first component, blood. If you see the blood cells are there, plasma are there, and free drug. So it can bind to both the sides, either to the blood or to plasma. Or the free drug can move to the liver, combine with the enzymes in the liver, form metabolite which are excreted, or from a protein drug binding in the liver. Now when the same drug crosses and goes across to extravascular tissues, then they can bind to the protein and form a tissue localization of drugs, or they can bind to the receptors where we can see in the tissues a pharmacological response. Now the same drug goes and binds to the kidney. If a drug goes inside the kidney, then first it finds a protein and binds to that. This is drug binding to renal tissues happens. Now if there is a carrier molecule, drug and carrier molecule gets active secretions and then they undergo elimination in the case of urine. So this is excreted in urine. So from the kidney, either the drug directly or with the help of carrier when they combine, they get into excretion through urine. Same way, in the liver also, when drug and enzymes form metabolites, they also undergo excretion in the urine. So this is a figure which explains you about how the binding of drug disposition and a clinical response occurs because of protein drug binding. Now, we will see binding of drug to blood components. Following entry of a drug into systemic circulation, the first thing which it can interact are blood components like plasma proteins, blood cells, hemoglobin. The main interaction of drug in blood component is with plasma protein, which is present in abundant. So it's an abundant amount, good amount is present, and in large variety, the binding is always reversible. So the first thing we have learned is a plasma protein binding. This can be due to hemoglobin, plasma proteins, blood cells where they come and bind, and there will be in large amount. The binding will also be reversible. Now binding of drug to human serum albumin. HSA we call it as, has a molecular weight of 65,000. It is the most abundant plasma protein, 59% of total plasma and 3.5 to 5% grams percentage. Due to the large drug binding capacity, human serum albumin can bind to several components having varied structure. This protein, human serum albumin, is present in abundant, and majority of drugs bind to human serum albumin. Binding of drugs to alpha 1 acid glycoprotein or AAG, alpha acid glycoprotein. This is an organosid. It has a molecular weight of 44,000 and the plasma concentration ranges from 0.04 to 0.1 gram percentage. It binds to the number of basic drugs like imipramine, amitriptyline, nortriptyline, lidocaine, propranolol, quinidine, and disopramide. Now we will see the binding of drug to lipoproteins. Binding of drug to human serum albumin and to alpha acid glycoprotein involves hydrophobic bonds. Since only lipophilic drugs can undergo hydrophobic bond, lipoproteins can also bind to such drugs because of their high lipid content. However, plasma concentration of lipoprotein is much less as compared to human serum albumin and alpha acid glycoprotein. Now we will see the binding of drugs to globulin. Several plasma globulins has been identified and are labeled as alpha 1, alpha 2, globulin 1, globulin 2 and gamma globulin. Alpha, beta and gamma. Alpha 1 globulin, also called as transcortin or CBG, corticosteroid binding globulin. It binds to number of steroidal drugs such as cortisone and prednisolone. It also binds to thyroxine and cyanocobalamin. Alpha 2 globulin are also known as ceruloplasmin. It binds to vitamin A, D, E, and K and cupric ions. Beta 2 globulin binds to carotenoids. Beta 1 globulins are one which is present, are called as transferrin. It binds to ferrous ions. So iron, ferrous, whatever it binds, it is called as beta 1 globulin. Gamma globulin, binding specificity to antigens. So gamma globulin are the one which always get together and binds to antigens. Now we will see binding of drugs to blood cells. More than 40% of blood compromises of blood cells, of which the major cell component is RBC. RBC constitutes 95% of total blood cells. Thus, significant RBC drug binding is possible. The red cell is 50 times in diameter as the major plasma protein binding component, albumin. RBC comprises of three components, each of which can bind to drugs: hemoglobin, carbonic anhydrase, cell membrane. So these are the things which are present in the blood cells. It can either be binding to hemoglobin, carbonic anhydrase, or to the cell membrane. Now, tissue binding of drugs. So normal tissue localization of drugs takes place. Now the body tissues, apart from human serum albumin, compromises 40% of body weight, which is 100 times that of human serum albumin. Hence, tissue binding is much more significant than thought to be. The majority of drugs that bind to extravascular tissues, the order of binding always will be the one which is having highly perfused organs. So when we say highly perfused, low perfused and medium perfused. So whenever we say highly perfused, the organ first which will come will be liver, kidney, lungs, muscles. Heart will be in a highly perfused organ. So binding of the drugs or tissue localization of drugs will always takes place first in liver, then in kidney, lungs, and then it will go to the muscles. Now, coming to the determination of protein binding. So there are various methods how we will determine the protein binding, and it is divided into two classes. One we say as indirect technique and one we say as direct technique. So what exactly it means is how to evaluate the protein binding has happened or not. So what we can do is we can take instrumental analysis, biochemical pathways. We can remove the blood samples from the blood, from the serum, from the plasma. We can easily determine what exactly is the drug which is binded and what is not bound. So instrumentation level, whenever we say, first the blood has to be withdrawn from the body. After the blood has to be withdrawn, the blood plasma, serum and the blood component, either we can take the whole sample of the blood and estimate how much is the protein binding of the drug taken place, or we can take the serum, because serum also contains majority of enzymes in which the drugs can bind. So there can be serum which can be directly bounded, then plasma proteins are there in which the drugs can bind. So these all things along with the analytical methods, we can use UV, HPLC or any other biochemical pathway for determining the studies of protein drug binding. So this is one way of indirect technique. So what exactly is an indirect technique, we'll see. Indirect technique is the one based on the separation of bound form from the free molecules, micromolecule. These techniques usually applied in biological samples like blood, serum, plasma for determining of the percentage of binding. Indirect analytical methods used in protein binding are dialysis, in which we will take a dialysis membrane, and dialysis membrane it will be present human serum albumin. Then we'll put the drug and see how much of the protein has been bounded. So protein studies are done by dialysis with the help of a dialysis membrane and then using human serum albumin solution. Dynamic dialysis, in this we are taking a membrane and directly we are doing dialysis, in which the molecular weight proteins or proteins which are bound high molecular weight will filter out, dialysis will be done, and then we can estimate the amount which is present. Or by ultrafiltration. Ultrafiltration is also a technique where we can filter the things and see the protein binding has taken place or not. Diafiltration, ultracentrifugation. Centrifugations will always settle down the protein bound complexes and easy for the evaluation will take place. One more method we call it as gel filtration. Gel filtration is one more technique where we can do the estimation of the indirect analysis. Now, coming to direct techniques. Direct techniques are those not required for separation. So when we say about separation, bond of form of drug and the free micromolecule, these methods are used for the estimation of number and elucidation of the characteristics of binding sites in pure aqueous solution of protein. Direct techniques used in protein binding are UV spectroscopy. In this, what we will do is we will have a particular wavelength and then they will put the sample which is protein bound and we will make it to a affected wavelength and then estimate the amount which is present. Fluorimetry, ion selective electrodes, these are all the techniques in which we can see how the protein binding has occurred. So the differences of all this has been taken by Shagil, applied biopharmaceutics and pharmacokinetics textbook. Then we have taken Brahmankar and Jaiswal, pharmacokinetics, Vallabh Prakashan. From this we have taken the reference. So today what we have learned, what is the mechanism of protein binding, how the protein binding occurs, what are the major sites of protein binding or how the protein binding takes place. Then protein binding, we have seen how to estimate direct and indirect method, two methods in which we are estimating the amount of protein binding in the drug, of the protein and the drug. So that's all for now. Have a nice day. Keep studying. Take care. Thank you.